Welcome to Climbing the Immune Ladder, Steps for Success in Science, the iiSIAR podcast, the show where we explore inspiring experiences in science and the untold stories shaping our community. Today, we will be your hosts, Salma, postdoctoral fellow from the University of Montreal, and
Janine Lückgen, senior postdoc in immune-mediated inflammatory diseases.
In each episode, we will dive deep into conversations with experts on innate immunity and in-cell inflammation, autoimmunity, and their resolution to bring you insights, ideas, and inspiration you won't hear anywhere else. Whether you're tuning in during your morning coffee or while doing your lab bench work, get ready to learn, laugh, and see the world from a new perspective. Let's get started. Today's expert is Professor Andres Hidalgo from Yale University. Thanks for joining us and welcome to our podcast.
Thank you for having me.
Could you please tell us a bit more about your line of research?
We immunologists, but I guess a typical type of immunologists. We study more physiology than actual immune cells. So with the excuse of chasing neutrophils and macrophages, which are two of the main types of innate immune cells, we just explore what they do in every corner of the body. So we end up being more physiologists, in my opinion, using the tools of immunologists. So the examples that you might want to hear about is we study circadian rhythms that are controlled by immune cells. We study the structure of tissues like the skin that are controlled by neutrophils, or cardiac function is regulated by macrophages, bone synthesis, and then these kind of not very immune questions, but mediated by immune cells.
And could you briefly let us know about your career trajectory in science?
Yeah, just briefly, I'm from Spain. I'm born and raised in Madrid, and then I did my PhD there in the 1990s. Sounds like centuries ago. And yeah, I started working on aspects of cell adhesion in hematopoietic progenitors. And then I moved, so I got my PhD from Universidad Autónoma in Madrid. And then I moved to New York City to do a postdoc with Paul Fernet at Mount Sinai School of Medicine. I spent 9 juicy years with him. And then when I thought I would stay in the US, I had a plot twist and I moved back to Spain for a position at CNIC, is the National Center for Cardiovascular Research in Madrid also. So my mom was very happy. And but I brought my then girlfriend, who was American, so that's why it was a bit unexpected that I would go back. But I was happy I went back. I spent 14 years at CNIC, and then another plot twist was in 2022 after COVID, and with a little girl with us, we decided to move back to the US, and I'm now a professor at Yale University.
What interesting pathway. Talking about the field of inflammation and immunology, what brought you to this field?
I think it was a bit serendipitous. This was not planned at all. But I've been always curious about science and research. And when I started my PhD and working on cell adhesion, all cells adhere to something, but we were particularly interested in fast regulation of cell adhesion, and the cells that do that the best are immune cells, lymphocytes, well, stem cells in the bone marrow too, but hematopoietic progenitors, lymphocytes, monocytes, neutrophils. So that was a bit the unexpected path that I became more and more interested in immune cells for based on that initial exposure to science, and then when I moved to New York to do a postdoc, I started doing intraocular microscopy in the lab of Paul Fernet, and that was a bit of a mind-blowing experience. I love to see cells under the microscope in living animals, and there was no turning back, right? So I got stuck with neutrophils rolling on vessels, and then I decided to chase them outside of the vessels. But that was a bit the path, and then more and more questions came about. So it was, I guess that's almost true for everyone. I don't think anybody plans when you're 23, 24 and you start your PhD, what was your career path. You just go with the flow. You like the questions. The more you know about something, the more questions that come up. So you just get sort of stuck in that area.
You were just telling us about this mind-blowing experience in the lab. Would you say this was a pivotal moment that shaped your journey, or are there any other moments that you can think of, either planned or serendipitous?
I guess that was a big one, because it was love at first sight, and literally first sight, because I was looking under the scope. I think you start realizing at that point that this is not a theoretical job. It's very much dealing with living organisms, living cells, and things that cannot be predicted to some point. And I think in my mind then was like, wow. But I also like the thinking process, and I think that was just naturally growing in me. And I suspect I would have had in other areas I would have had similar experiences, just because biology is so cool and there's so many turns and interesting things. So there's nothing planned about me. Let me put it that way, and about my career.
And beyond scientific curiosity, is there any personal motivation that has driven your work in immunology?
Well, fortunately not. Although the more you get exposed to this, you end up being exposed to to the stories of people who suffer conditions, cancer, autoimmunity, septic shock. You can think of family or the family of friends. Not me or my immediate relatives, thankfully. So the motivation for me has been purely intellectual, and I'm not saying that that can change at some point. Use the tools that I have to make a difference in specific disease conditions, but I've always thought that if you're not driven by curiosity, if you're driven by a mission to cure something. You can make a difference, of course, but I think there has to be some innate motivation. I want to cure cancer, of course, but I realize this is not something that happens overnight. I'd rather be interested in what happens to this type of cell, and why does it do this thing, and how can we control this? It's a little bit like an engineer likes to build machines. It's a bit the same thing for me.
During your career, you made many very important discoveries, especially regarding neutrophils and the circadian rhythm. Could you highlight a key discovery or project you're most proud of and how it impacted the field?
Yeah, well, I'm proud of almost everything, like I said, because it's driven by curiosity. So when you find something, you say yes. The hypothesis was true, and we are the first ones to have realized this. Even if it's a small finding, it gets you a little bit closer. But having said this, I guess a turning point, and I'm proud of it, perhaps because it was the best, the first. More substantial discovery in the lab was when we were working with my first PhD student, Maria Casanova, and we were looking into the biology of neutrophils in blood. So very blank question, so to speak. And we had this observation taking blood samples and seeing that how tight were the oscillations in the change of neutrophils in blood, right? And then starting asking questions if it was only numbers, it was the phenotype, and then we found specific markers that could define that there were also some molecular changes on the cells at the same time. It's not just numbers as they're being released, which was what was described at the time. And then I guess the first challenge was and something I learned from my postdoc mentor, Paul. So who cares about this, right? So but we have an observation. Is this important for anything other than for us to publish a paper? What is it doing in the body? And so we started looking at the bone marrow. Maria was able to tease out this coordination between the cells that are dying, the neutrophils that are dying naturally in the bone marrow, and they get their specific times of the day, and then the release of new cells from the bone marrow. So it's kind of realized that there was this very cool coordination between two systems, the immune system and the hematopoietic system, was quite enlightening, and in fact, it not only allows us to make a significant contribution that people apparently like, because it's quite excited, this coordination, this circadian coordination between the two systems. It also opened a field of research for us that we still work on, which is like circadian rhythm. So in the end, is physiology. How do you control the bone marrow output? So that's something that we look in many other organs now. How neutrophils function as relay signals, a little bit like hormones in a way, but they're cells. So it's a bit more complex than hormones, but the principle is the same thing. They get into tissue, they tell something to that tissue, and then the tissue starts acting accordingly. And the way it acts it can be quite surprising at times, depending on the tissue.
And we've been talking about scientific questions. Do you have any process or tip you would like to share with us to identify this high impact research question?
That's the million dollar question. I guess there's a process. I'm not very sure what the process is you develop an instinct into all these questions. It's literally, I guess this is to reach to everyone, where you say something is cool, or this is weird, or this is cool. And what's the definition of cool? That changes with everyone. But a cool can be something just simply very unexpected. I'll give you an example of a recent study we did in the skin, and we were doing this profiling of what happens to tissues when you deplete neutrophils, right? So it's a very exploratory question, and we'd like to do a lot of these screening tests in the body. You deplete one cell, what happens to the tissue? You add the cells, what happens to the tissue? It's a bit of a broad fishing question. And then when you see something like, you look in the bone marrow, the changes in the bone marrow, but the bone marrow has 50% of the cells in the bone marrow in the mouse, they're the neutrophils, so that's not very unexpected. But then you look in the skin and you see that the changes are consistently associated with changes in the extracellular matrix genes. And this comprises many, not only collagens and laminins and all those things, but also enzymes that regulate the extracellular matrix. And you say that's odd, right? That's weird. Is it possible that there's a connection of neutrophils regulating the extracellular matrix? So of course there's two components to this. One is neutrophils degrade extracellular matrix. That's been known for decades. So that's not very good. That's not something I would pursue. But the differences was when we look a little bit into in more detail into the question, that when we depleted neutrophils, there was less extracellular matrix, no more. And that's not what you expect, right? So you start asking the question, wouldn't it be cool if neutrophils were building matrix? In part because, one of the experiments was to look at the cells and see that they was collagen inside of them. That could be anything. That could be neutrophils eating collagen as a part of the degradation process, but that could also be neutrophils producing collagen. So we decided to take the unexpected observation and build up on that. So that's a process. There's no clear recipe. It's just following your nose and asking bold questions, many of which are just wrong and many of which are right. So, you just go for, and you leave questions behind that would be interesting. That would be interesting for a small paper, small observation to advance what others have done. But isn't it more fun to do things that are completely new? So we tend to go for the risky part, and when we're successful, then it's really, really cool.
Good advice. So a scientific journey is usually full of challenges. What strategies help you push through the toughest scientific challenges?
Oh, that's a tough one. Some challenges you just simply cannot solve. Challenges associated with funding for a specific project, or making a student or a postdoc successful. There's this stories of success and stories of frustration. Um, so I think this is a personality thing. It's a very personal question how you deal with defeat. There's something I think is intrinsic to each person, and there's something that comes from experience. Um, One thing that you learn is that the next day will be better. I learned that during my postdoc when you have a very frustrating week, and then you say, "Shit, I don't want to do this anymore." I guess one trick or one trait of the personality that helps is when your brain is always looking for the path forward rather than dwelling into the frustration. Right? You say, "Well, this was frustrating. Take a few hours, just deal with it." And the next day, think about, well, it was just a bad result, a bad week. Next week will be better. It's a personality thing, you guys, not something that you can define in yourself. But when you learn that this works and your brain acts by always trying to move forward rather than getting stuck, then I think that that's worked for me.
Could you share with us any crucial decision or opportunity you had that significantly advanced your career?
Yeah, I guess there's a couple of points that, in retrospect, were great. One was moving to New York at the time. My PhD hadn't been very successful, so I didn't have much to account for in terms of publications. So it was tough search for a position, and I tried several. And then I wrote Paul Fournette. I mentioned him a few times, and he was a junior PI at the time, and so he took me in, and that was the best thing that could happen to me. I love New York City. I love Mount Sinai. I love his lab. I liked him a lot, and the projects, little by little, started coming together. So that was I don't know if it was luck or what it was, but that was a defining moment. I often wonder if what would have happened in another place, another PI. And the second one was moving back to Spain. You know, Back then all the science was in the US, and Spain in particular was not a particularly strong spot in science. But there was in this new institute they opened a couple of years before I joined. I joined in 2008, and they opened in 2006, I believe. And things were great for me there because the structure, the people, the facilities, everything was good. The PIs, and that's I always emphasize to people when from my lab when they get independent. But I usually tell them is take your time to find the place that works for you. In my case, I mean, I thought about and I look at the places, so it's not like I just went straight. But it was a risky move because there was not a new place. You don't know how the leadership is going to be there, how long the money is going to be supporting you. But it worked. The place has been great, and it's still a great place for research. Great facilities, get support, intramural funding. And so I guess, when you make, I won't say two good decisions because they were both risky. But when you're lucky in two consecutive steps, then it's difficult to fail.
Talking about risks, how do you evaluate and seize new opportunities while managing existing commitments?
The trick is you just do the best you can. It's to try to be fair and cautious. Um, I'm always very careful with commitments that I have. I try to treat collaborators, for example, or people in the lab fairly and responsibly. So, if I'm guiding somebody in a PhD, then I'm committed to that. If I have a collaboration, then I'm committed to that. So I think being really respectful, then you get respect back. and that helps you along the way. When people think that you're someone reliable, that's the best thing that can happen to you. So you have to pay back, right? You have to be always respectful with the commitments that you have. It doesn't matter how big or small they are.
And what non-scientific skills have been most critical to your career progression, and how did you develop them?
I guess what I mentioned, this sense of responsibility. When you commit to something, be careful and treat people as you would like to be treated. So that's something that is helpful. It comes also from a sense of like how you're educated. So I won't say that you are the way you are, but if you can work on just being responsible and respectful with everyone, that helps. It's how to advise, how to deal with personalities, with results, with frustrations. It comes from experience and keep it cool. So again, you learn this with time. It's a bit of an intuition of how you should do things. Obviously, you just don't go out screaming to the lab when you're frustrated and start firing people. So that's an example of a. And you would be surprised. There's people who react a little bit like that, or threatening all the time, or just giving a hard time to people constantly because you're frustrated, right? So you need to let it go. So you learn a bit the psychology of how to handle these things for yourself and how to try to help people on the other side. That I find more tricky, and I'm still learning a lot on how to deal with different personalities. People who are super driven and super gifted, that's not a challenge. But people who are clinically having problems, then that's a bit of a challenge. How do you deal with that? What happens if you talk to them, you implement changes, and then they work? That's wonderful, but often they don't work. So what do you do next, right? Should I ask this person to go? Or should I just do something to support this person? But also that when you make a decision, you have to go along with that. Sometimes it's a good one. Sometimes it's not such a good one. Um, so yeah, you learn to look at the big picture and try not to get tracked by emotions or personal likings or things like that.
Learning is always part of science. And but how do you stay current with scientific and technological advancements that continuously foster your personal growth?
A lot comes from seminars. Now that I have less time to read, now of course reading papers, that's a big source. But going to seminars and often also just discussing with people in the lab what they've seen. So one thing that I learned from a friend quite a few years ago is that if you read too much and go too dogmatic by learning everything that is in the textbooks and the papers, then you don't learn to think for yourself. Like you have to follow the data, especially when you go into a new field and everything is new for you. You have to go a little bit with your gut feeling, learn really the basics, read a couple of reviews of papers that are relevant, but then follow your instinct. As an immunologist, when we get into a lot of fields, there's nothing done from our perspective, but there's a big field on the other side that tends to be very dogmatic
and overlooked, yet essential for success, both in science overall and specifically in this area of immunology.
Yeah, immunology or any field, I don't think that changes from one field to another. It's being resilient, dealing with frustrations in a productive way. That's the most important thing. If you like or you don't like science, you realize soon enough you don't like it, you should leave it. But if you stay, right, that's important. I'm not saying that science is the ultimate goal. You have to do that no matter what. You don't have to do that if you don't feel that that's your thing. But if it's your thing, there's going to be good days and bad days, boring days and super exciting days. Time is a healer, and I think one can work in getting a positive attitude into how do I solve this, and that's the fun part of science too, right? Is problem solution. Try to be creative with that thing, or try to find tricks to do it.
Have you ever received from your colleagues or a mentor, probably, a piece of career advice that you would like to transmit to new generation?
Paul used to say, well, whenever we came out with some results, say, so who cares? Who cares sounds a bit like a bad approach to this. But when you got to know him, it meant, okay, show me the relevance of this. You need to think about the impact of what you're doing. So I guess what I learned is that you have to set a focal point. You know your reader, you know where you're going, and you know where you are. It's so much easier to draw a path and to build a project when you know where you need to go, rather than just explore without really knowing where you're going. So that was a useful thing that I've learned from a mentor, to set the end point as soon as you can.
And now, what advice would you give to somebody who is just starting in the field?
Yeah, I don't know if I'm able to give any advice, but from all the things that I said, I think it's quite clear that first you need to decide if this kind of job or this kind of profession or lifestyle is right for you. And there's many hints in which you should sense this. Are you excited when you go to lab? Do you like to read papers? Do you like the vibe of the lab? Do you like to be in the bench and doing things, work with animals, work with cells? Do you enjoy that process? If you don't, piece of advice, don't just because you're getting a salary, just don't continue down that path, because that's not going to be good for you unless you want to be I don't know, making a living is an honest thing. It's not like, anybody can give up a job. But if you're looking for a profession, something you want to do for life, then just don't get stuck to something just because you studied biology and you get a PhD position with a salary. That's not good enough reason. If you decide you want to move on, then there's things like I mentioned, depending on your work, finding the right mentor, again, is a piece of luck. It's not something that you decide often. That helps you helps you with navigating your early professional career. If you do physiology, immunology, it's something that's been very useful for me is think about where your project is going. Try to answer that question first. What is the relevance of what I'm doing in biology? And that's very interesting and important, because I think it makes you think about the whole process, not just the next experiment. You need a bit of a vision to do the better science, I think, right? You need to be part of a trajectory. And so that's important, that you understand science as a trajectory, as a process. I'm here and I want to get here. So try to define that. And then there's a lot of talk about mental issues and dealing with frustrations, things that change with each generation. So learn how to live with how to deal with frustrations. If you cannot deal with the frustrations shared with the lab, maybe this is not your job. But in life, it would be a great thing if you learn how to deal with the hurdles that you will find.
How do you navigate the demands on high-profile scientific career while maintaining a healthy work-life balance?
Again, the way I deal with it is enjoying what I do. I like it so much that I don't find this a burden or struggle to do this. I work a lot and I think a lot about what I do. We have a little girl now, so it makes it a little bit more challenging because she doesn't care about immune cells at all yet. Nothing needs to be done right away, but you need to feel that when you live every day, you've accomplished something, and you have to have this sense of responsibility that you finish and stuff, complete things. Once you have that feeling, there's no time constraints, right? You take a week off, but you got the stuff that you wanted to do done. You come back and you continue. You're working for yourself. So once you assume that, I don't think I never felt an issue of work-life balance. Some days I'm very tired, so I go rest, and the next day I I take off, or I go on vacation. But when I'm here, I'm just always thinking of things I want to do, accomplish, learn, and that's satisfying for me. So I've never, fortunately, haven't had this struggle, and I know that's not something that you can tell to everyone, but it's a question of attitude. It's always been a question of attitude. There's no an issue with balance once you know what you are trying to do. Is your project. If you don't enjoy the project, then there's an issue of work-life balance. If you feel that you're in the lab, but you really want to be with your partner, or going to a terrace, like in Madrid, we do this a lot, for to have a few beers and hang out with friends. If that you have that constant feeling, then you shouldn't do that thing. The balance is just when you're pushing yourself to do something you don't want to do. If that's what you want to do, I don't think that there's a balance issue.
Yeah, exactly. When you love what you do, it doesn't really feel like work, right?
Exactly. You're accomplishing something.
And what challenges and which opportunities you see for the next generation of scientists?
Mm. Well, I think there's a big challenge in how science is supported by society. I see that this is a big change coming in the way science is being made for several reasons. One is right now this loss of support that I sense from government. Science is lost a little bit the edge of being driven by curiosity and by the job that I was mentioning before, and it's become more like an established 9 to 5 structure. So it dilutes a little bit the passion and the vision from the job, right? Everything has become so structured that is difficult to cope with some of the requirements of the job. You are paid to be curious and to discover. But there's been so much a structure of what you should do and what you shouldn't do, the time constraint, the work-life balance discussion, the in Spain, a place where I was working, they said this limit of 7 hours per day to work to the students and postdocs, which I thought it sounds aberrant, I think, for everyone, but that came and that's been imposed by the government, in an attempt to protect the workers. They say, okay, now you scientists are in the same category, and that doesn't make any sense. You realize 7 hours a day, some days you can do with 6, but most days you will need more depending on the experiment you're doing. And you want to, like I say, you have a sense of accomplishment. You want to do things. If you have to leave at 5 because otherwise you get in trouble, so what kind of, right? So that over structuring this job, I think it's a challenge how society and this discussion of work-life balance and hyper protection and employee, it's a bit at odds with people who seek their own path and passion. So that's one opinion. The funding is always going to be an issue, and I think governments are backing off little by little from being the main supporters. And the good thing is private funds in some countries are taking over, so that there will be a change in how this is supported. And then the technologies, the way science is done now is you need more to be a computational biologist than a bench people. I love bench, and I think if you think when you're doing the actual experiments, the crafting of working with your hands and doing the discovery when you develop a Western blot or whatever you're doing. So that's changing a little bit. There's a lot of leaving your thoughts to the computer rather than doing the thinking by yourself. So that will be a challenge in how we will deal with so much mega data coming from. How do you deal with it without getting eaten by the computational aspect of it? Computers are great, but if they do the thinking for you, that can be an issue. So I think that's something people will have to learn dealing with in the next generation.
Before we come to an end, we would love to hear a standout moment from your career, perhaps something lighthearted, surprising, or out of the ordinary that you feel comfortable sharing with us.
I love when I meet trainees after a while, people who work with me, and some of them are good friends to this day. So I love that and reuniting with them. I love the new friends that I've gotten, close friends that I've gotten through this job. Right, you share this passion and the questions, and you get to meet with them. I love when we get a paper accepted. We just had a paper accepted 1 hour ago. A pretty big deal. We've been working with this for a long time. So an hour ago, that it's the culmination of several years, quite a few people, and this the first author actually of this paper. Every paper is a bit of, how this goes, a bit of a torture. You need more experiments, but why do we need to do this experiment? Because the reviewers has just do it. Okay, but it's stupid. Okay, but just do it. So it's a long process from the discovery to then the publication process, depending in what journal. And then finally you get to the end and you get to give a hack to the person to say, there's now you discover how you go from alpha to omega, and this is what this process is like, getting things done and discovering. In this particular case, the first author, when we were in the middle of the experiments, he was diagnosed with leukemia, and that was in March. And she just came out. Fortunately, everything went well of that, like a month ago, right, with all the sessions, and the diagnosis was good. So hopefully she's cured now. But, she's still with a very short hair after all the chemotherapy. So, so she came back to the lab after 7 months last week. And when you leave, you don't know if you're coming back at all. So that's talking about dealing with issues in the lab. But anyway, she came back, and today we got the news that her paper was accepted in Nature. So that's a great moment too.
Congratulations for your new paper. We're eager to read it.
Thank you. Hopefully soon, after the last editing.
That's a wrap for today's episode of Climbing the Immune Ladder, Steps for Success in Science, iiSIAR podcast. A huge thank you to Andres Hidalgo for sharing his insights, stories, and experiences with us. We hope you found this conversation as inspiring and thought-provoking as we did. If you enjoyed this episode, don't forget to join us on the next episode and share it with anyone who might benefit from it. You can also follow us on our webpage to stay updated on future episodes and innate immunity updates. Thank you for listening, and remember, persistence and asking the right questions are what drive science, and maybe even your own journey. Until next time, stay curious.